Pediatric acute leukemia can be broadly divided into three categories based on the respective lineages: acute myeloid leukemia, or AML, acute lymphoblastic leukemia, which is further subdivided into T cell and B cell categories. Now, lineage specification requires flow cytometry and on peripheral blood or bone marrow samples. But a comprehensive diagnosis of acute leukemia requires much more. But because of this, comprehensive diagnostic requirements, WHO has introduced the category of NFC, or not further classified, for BALL in resource-limited settings, because lineage specification is the most important diagnostic decision. But it still falls short of risk identification or the opportunity for targeted therapies which can drastically improve the prognosis of children with cancer. So, as I just said, a comprehensive diagnosis requires a set of different techniques, including flow cytometry, but then there is also FISH, karyotyping, PCR, and sequencing. And all of these techniques are required for a diverse set of abnormalities like translocations, copy number changes, ploidy changes, transcription profiles, point mutations, and then translocations. And all of these together are currently required for a comprehensive acute leukemia diagnosis. And all of these techniques for us are add-ons rather than replacements, and these techniques just keep on adding the burden of diagnosis that is required even in low income settings. So, and this is the reason that specialized cancer diagnostics have a very low penetrance in low and middle income countries. So some of the obvious reasons are cost, technical capacity, but there are also issues with centralization because of transport and logistic issues, and then also supply chain issues. And so because of these problems, it is estimated that in Pakistan, less than 50% of pediatric cancer patients receive a proper diagnosis, and this is just talking about lineage specification. So against this background, we asked the question together with our collaborators if we can have a single technique that can unify cancer diagnosis. And we think that long-read transcriptome sequencing is one such technique. So just to show some of our data. Before that, I wanted to show this slide where, which explains how we are currently using long-read sequencing to establish its accuracy against flow cytometry. So we are using an ML classifier developed by Dr. Jeremy Wang's lab at UNC. So the classifier gives lineage together with the confidence threshold. And so if the confidence level is above 0.8, we call it a confident call. Now, and if it's below 0.8, we call it a test failure, which means that it either needs to be repeated or it has to reflex to flow cytometry. So if the confidence call is above 0.8 and it is concordant with flow cytometry, then it's an accurate call. And if it's not concordant with flow cytometry, then it's an inaccurate call. And so before moving further, I also wanted to show this slide, just showing that we've used two different sets of nanopore flow cells and kits, the previous version and the new version. And understandably, the total throughput increases with pore count. But with the newer version and the of kits and flow cells shown in orange, we have a much higher throughput than expected for the pore count. And so that is why we've divided our data based on the kit version, and the flow cell version. So these are the results from the previous flow cells and kits. So we did a total of 258 samples using those kits, and these slides just show the discordance and concordance rate for the different types of leukemias. So in total, we got a discordance of 8%. It was the lowest for B-ALL, just 3%. For AML, it was 6%, and for TLL, it was 16%. So with the newer version of kit and flow cells, we got somewhat improved results, a lower discordance, a total of 6%, and then for AML it was just 4%, 2% for BLL, and for TLL it was 15%. So just to place these rates of discordance in some context, so I looked up the concordance of two standard techniques that are used for immunophenotyping, so immunohistochemistry and flow cytometry for different kinds of cancers. And we saw like these are two gold standard techniques, and in these three publications you can see that there's a discordance ranging from between 9 and 20% between two gold standard techniques. So because we are trying to do clinical validation apart from accuracy, we also have to determine precision or reproducibility as well as the limit of detection. So we got more than 90% concordance repeating the samples with different types of, with different machines and different staff and using different sample sources. And then we also got a threshold of the total number of reads at 10,000. So even with 10,000 reads, you can get accurate calls. And then we also try to determine the minimal blast percentage that can give an accurate result. So this was around about 40% for all leukemias, but it was much lower for B-ALL. Because in the first table, you can see that we ran several blood and bone marrow pairs with different blast percentages. And in the blood, so even for a percentage as low as 2%, we were able to get an accurate call. But with TLL, again, with the bone marrow and peripheral blood pairs with different blast percentages, we got a test failure with 9% blasts. And for AML also, it was variable. These are all samples for which these were different pairs of samples that had different blast percentages. So sometimes we could get an accurate call with a percentage as low as 18%, but not all the time. So I think the threshold at which we always get a concordant, confident call is around 40%. So we do have some current limitation, one of which is mixed phenotype acute leukemias. We just don't have enough samples for that. These are rare. And then also non-leukemias. So we need to work more on training the model to be able to detect non-leukemias accurately in the sense that to call it a test failure. So this is some ongoing work in order to be able to bring it to the clinic. Not only do we need to cover all of the parameters that are required for clinical validation, we also need to determine the accuracy of this model for other kinds of tests, including copy number variations, translocations, point mutations, and transcription profiles. So currently, the data that I've just shown, it was low depth shallow sequencing using MinION, which cost around about $80 to $100 per test. But to be able to include all of the different kinds of abnormalities in a single test, we will definitely need to do a high depth transcriptome sequencing. And for that, we plan to do the next set of experiments using a P2 solo. And so I've just created two scenarios because we don't know what will be the threshold depth at which we will be able to accurately call all of the other types of variations. And so in our setting, even if we do a high depth sequencing, the cost is around about $600, which is much less than the cost that we currently have for all of the other tests combined. And I think in summary, So diagnostic deficiencies are a barrier to cancer treatment in many countries, including ours. And then ONT mRNA expression profile has the potential to accurately do lineage specification, but we still need to do extensive validation, especially in our clinical lab, to be able to introduce it as a clinical test. So I would like to acknowledge our collaborators, Jeremy Wang, Thomas Alexander, and Nikhil Bhakta. It was a collaborative work, and thank you so much.